Hello everyone. In this narrated PowerPoint lecture, we will conclude our review of Chapter 1 by discussing Section 1.4, What is Science? Let's begin. Science is the systematic inquiry through observation and experiment into the origins, structure, and behavior of living and non-living environments. Literally, the Latin translation of science means to know. First, we will talk about the nature of science. Then we will talk about some ways in which science is conducted. The nature of science, often abbreviated NOS, N-O-S. Note, this material is not in your textbook. When we talk about the essence of science, we talk about what science is and what it isn't. Almost as if science has a personality. What is the essence of science? Science is tentative. This means scientific knowledge can change. What was true at one point may no longer be true once new information and new technologies become available. Science is creative. Science involves the use of imagination. Science is subjective. Scientists viewing the data can develop different conclusions due to personal experience, background, and education. Empirically based. Science is verifiable or provable by means of observation or experiment. When we say something is empirically based, we mean it's based on data. Science is socially and culturally embedded. Science can affect society, and society can impact science. This is the essence of science. When practicing science, important distinctions are made between observation and inference. An observation is the factual information gathered from your senses. Examples are from touch, sight, or smell, hearing, and taste, or factual information gathered from extensions of our senses, such as with a microscope or a telescope. Inferences are interpretations of information taken in by our senses. There's also a distinction between theory and law. We'll talk about that distinction more in a future slide. The process of science. Scientific studies usually begin with a question, but not always. No single set of or sequence of steps is followed in all scientific investigations. Below is a list of 4 different ways in which science is commonly done. There's hypothesis testing, also known as the scientific method, discovery science, descriptive research, and correlational studies. The most commonly used method, and the method that we will talk about, is called hypothesis testing, also known as the scientific method. On this slide, I have listed all of the different steps, so to speak, that you would see performed when using the scientific method. Studies conducted with scientific method or hypothesis testing begin with observations. The researcher uses their five senses and instruments to make observations. These observations often lead to a research question. This research question can be developed based on observations, previous information, prior knowledge, and/or preliminary information. From this, the researcher will develop a hypothesis. This is a proposed explanation for a natural phenomena or educated guess. Then the scientist will make a prediction, which is the expected outcome if the hypothesis is right. The hypothesis and the prediction are used to develop a controlled experiment. When you conduct your experiment, you will gather data, you will analyze the data, and report the data as results. Then in your conclusion, you will talk about what the results mean, whether or not you accept or reject your hypothesis, and you might propose future experiments. Please be aware that your experiment and results must be repeatable by someone else. Let's look at an example where the scientific method is applied to a common problem. If the lights went out in your house, you would probably grab a flashlight. If the flashlight didn't work, how would you solve this problem? So your initial observation would be, my flashlight doesn't work. That leads to a question, what's wrong with my flashlight? Your hypothesis or educated guess might be the flashlight's batteries are dead. You predict that if I replace the batteries, the flashlight will work. Then you design a controlled experiment. And in this case, your experiment would be to replace the batteries with new ones. Then you would attempt to turn the flashlight on. If the flashlight now works, the experiment supported your hypothesis, and you might make some additional predictions on what flashlights need to work. If replacing the batteries did not work, your hypothesis was not supported, and you might revise or come up with a new one. Some alternative hypotheses could include a faulty bulb, faulty wiring, or defective battery. When trying to solve problems such as this in everyday life, you don't typically refer to the thought process as the scientific method or hypothesis testing. However, the same logic and reasoning that is used to solve problems such as this one is the same logic and reasoning process that would be used in a scientific study. Let's look at an example of hypothesis testing, also known as the scientific method, in a scientific study. In this study, the scientific method is used to determine whether or not a new artificial sweetener called sweetener S causes cancer in lab mice. Examples of artificial sweeteners include sucralose and sweetenlo. In this study, the researcher used two groups of mice, the control group, which received food with no artificial sweetener, and the test groups, which received food with the artificial sweetener. The test groups were further divided into subgroups based on the percentage of sweetener in their food. The amounts used range from 10% to 50%. The researcher conducted the experiment twice, recorded the data as his results, and presented them graphically. In this study, can you identify the independent variable, dependent variable, and control variables in this experiment? What do the results show? What can we conclude about the sweetener? The answers can be found at the end of this presentation. If you are having trouble identifying the independent, dependent, and control variables, or understanding the difference between the control group and experimental group in the previously discussed study, let's take a look at some of the definitions of these terms. Any good scientific study will have these features in them. Control group versus experimental group. The control group is the subject or subjects that go through all of the steps of the experiment but is or are not exposed to the factor being tested. In the previous example, the control group would be the mice that had food that did not include the artificial sweetener. The experimental group, also known as the test group, is the group that is exposed to the factor being tested. In the previous study, the experimental group would be the mice that receive food with the artificial sweetener in the food. Independent versus dependent variable versus control variable. Independent variables are variables that are manipulated by the experimenter. Dependent variables are factors or outcomes measured from the subjects. Controlled variables are variables that the experimenter attempts to control so they won't affect the experiment or influence the results. Let's look at an example to help illustrate the difference between independent, dependent, and controlled variables. Let's say that you were interested in testing whether or not a new drug caused nausea and vomiting in people who took it. The independent variable would be the new drug, because you, as the experimenter, are manipulating which group will get the new drug. The dependent variable would be the amount of subjects that get nauseous or vomit. Nausea and vomit are the outcomes that you're looking for from the patients or subjects receiving the new drug. The independent variable is what you are controlling in the study and is also sometimes referred to as the test factor. And the dependent variable is the outcome that you're measuring or what you're going to be calculating or counting or detecting as a result of the test factor. Dependent variables are called dependent because they are dependent on the independent variable. This differs from controlled variables. Controlled variables are things in your study that you attempt to control so they won't influence the results. In the study that determines whether or not the new drug causes nausea or vomiting, some things that you would control would be maybe you would make sure all the subjects are the same age, the same sex, or maybe you control what they ate that day. You would make sure the two groups ate the same food or at the same time. When you do your scientific study, the only thing that should be different between your groups is that test factor. Everything else should be the same. Lastly, deductive versus inductive reasoning. These are two logic and reasoning styles that you can see illustrated when scientific studies are done. In inductive reasoning, lots of non-contradictory facts can lead to a broad generalization. With deductive reasoning, you go the other way in that a well-supported generalization is used to develop a specific set of ideas about a phenomenon. In this slide, we have an example of how inductive reasoning could be used in the development of scientific theories. For example, the theory that the Earth exerts gravitational forces on objects began from repeated observations of objects falling downward toward the Earth and from no observations of objects falling upward away from the Earth. With inductive reasoning, several isolated observations that are non-contradictory are used to arrive at a general idea, theory, or conclusion. With deductive reasoning, this is the process of generating hypotheses based on a well-supported generalization. For example, based on cell theory, any new discovered organism would be expected to be composed of cells. With deductive reasoning, the general theory is used to predict the various facts that should occur if the theory is true. Science is based on the principle that all events have natural causes. The belief that all events can be traced to natural causes that we can comprehend. This is referred to as natural causality. Science can't be used to test the supernatural. Anything tested in science must be testable and verifiable with the five senses or extensions of the five senses. Faith-based ideals are not testable and lie outside the boundaries of science. Lastly, let's talk about the difference between a theory and a law. In science, theories and laws are two very different, distinct bodies of knowledge. A scientific theory is a general and reliable explanation of important natural phenomena that has been developed through extensive and reproducible observations and experiments. Theories can be disproven if new evidence arises. Theories tell the how and why behind a natural phenomena. They are analogous to an inference in science. This differs from a scientific law, which is a statement of observed fact that has been demonstrated to be true in all situations. Laws tell what happens. They are analogous to an observation in science. Laws do not explain why. One common misconception seen in many science textbooks is that theories eventually turn into laws. This is not correct. Let's review. If an experiment is done to see if a dietary supplement affects the weight gain of chickens, what variables need to be controlled? A, where the chickens are kept. B, the availability of water. C, the temperature of the surroundings. Or D, all of the above. Please remember to write down the letter that corresponds to the correct answer, as you will need to input this in the lecture quiz for this module in Blackboard. Next question. An experiment is done to see if eating breakfast affects students' performance on exams. The independent variable is A, the student's scores on the exams, B, the age of the student, C, whether the student has breakfast or not, or D, whether the student drinks coffee or not. And this slide has the answers to the mice and sweetener S study. You were asked to identify the independent variable. The independent variable is the artificial sweetener. You could also say it's the amount of the artificial sweetener used. Identify the dependent variable. This would be the presence or absence of cancer in the mice, or the rate of cancer. Identify some control variables. Some control variables for that study would be the cage where the mice are kept, the type of food they eat, the type of mice, or even the sex of the mice. And lastly, what can we conclude about the artificial sweetener used? The particular sweetener used in that study appears to cause cancer in amounts greater than 10%. The rate of cancer increased as the amount used increased. The hypothesis was supported. This slide concludes the narrated PowerPoint lectures for Module 2 on the properties of life and scientific inquiry. Please complete the lecture quiz in Blackboard using the letter that corresponds to the correct answer for each question that was presented in this PowerPoint.